Hello and welcome back to the MCAT grind. We have Biochem Chapter 6, DNA and Biotech. Of this chapter, there's about 15% yield, and that means you will see a lot of questions on it. So whether it is in passage and asking you to determine what happens if this chemical stops this certain enzyme from helping this, or about histone questions. Common questions in discrete forms are also asking like the basic structures to know. So if you know adenine, guanine, cytosine, uracil, thymine, their structures by heart, then that's another 1.2 points for free. There's also common questions of asking about base pairing, and you just need to know, right, from biology. AT bond together, CG bind together, and CG uses 3 hydrogen bonds, whereas AT uses 2. Now, to remember structures, we have adenine. Adenine and guanine are both purines. Purines means they have 2 member chains in them. And adenine, specifically to remember it, I count 1, 2, 3, 4, 5. Adenine is, I think of this 4-member ring, plus add N on top. So it's adding another N on top, like adding some kind of scoop and confectionery on top. So to remember the rings, you would know that rings, the double bonds can only go every 2 space, right? They can't be next to each other. So 2 space, so 3 in ring, and this is the same as guanine, and they're both having 1 extra 5-member ring on the side. Now, only difference between adenine and guanine is that guanine has an O carbonyl group instead of the NH2 on the top. That amine group gets moved down to the bottom here instead. So guanine brought the N2 for a hike into Guam, right? Guam, where vacation spot, and amine is going there for a hike, right? See, adenine here doesn't have anything down here because there's no one in Guam, where guanine brought amine all the way down for vacation, and over there, carbonyl there, that spot is vacant. Cool. So those are purines, and they have 2 member rings. 1 6-member ring, 1 5-member ring. Pyrimidines only have 1 6-member ring. Now we have cytosine, uracil, and thymine. Cytosine and thymine are found in DNA. However, thymine isn't found in RNA, and it gets replaced with uracil instead. So, cytosine uses a basic structure, and as we saw, there is N on the top left and N on the straight bottom, and the same place for the 2 double bonds. However, just like guanine, where it's bonded to, or they all 3 of them have a double bond carbonyl on the bottom left. However, cytosine is the only one without the carbonyl O on the top because the O went inside the cytosine, right? Uracil and thymine, they don't have O in their name. Cytosine is the only one with O in the name, and that O was taken from the very top, the amine group. They took that O from there. Uracil is similar, but it's different from thymine because thymine has additional methyl group on the top right. I remember this because DNA is where things come from, and methyl is there because DNA is itself is a longer chain, and uracil is part of RNA, which is a smaller chain, so it's smaller. It doesn't have that methyl group. And you just need to know that guanine and cytosine go together, adenine and thymine go together, so that means guanine, for every guanine you have that equal amount of cytosine, and for every adenine you have equal amount of thymine. You can use this to determine by Char-Gav's rule that if you know a percentage of one of them, you know the percentage of any one of them. If you have 5% thymine in your DNA, then you also have 5% adenine in your DNA, and because that's 10% altogether, you have 90% for guanine and cytosine combined, so 45% each composition. Make sense? Next, we have sugar phosphate backbone. That is along the sides of the ladder. Remember, it's a double helix, right? So it will be A or any of these nitrogenous bases as the rungs, and on the side you have your backbone, which goes from phosphate being on the 5 and then going down to a 3, down to the sugar. We have ribose, which is itself a 5-carbon sugar. Then if you take away one of the O's on the fourth, I think the fourth alcohol, then it's deoxyribose, and that's what DNA comes from, deoxyribose. They took away one of the O's, and if it's deoxyribose, you took away both of the O's. The second one taken away from the third carbon of the sugar. And we have heterochromatin and euchromatin. Histones help wrap DNA, right? If all of your DNA in a single cell is stretched out, it stretches out to 2 meters. So you want a way to be able to ravel it, and a way to unravel it. So you ravel it around your histones, which H2A, H2B, H3, and H4, with H1 being like a little clip to keep it in there. And what happens is the part that gets wounded together, you can think of it as an extension cord you're not using at the moment, right? And at the times that you need to use extension cord, you need to take it apart and use it. While it's still wound around, the DNA is silenced because you can't access it. It's being wound, and that's heterochromatin, silenced. Euchromatin, EU chromatin, is the long parts that are able to be accessed by polymerases, primers, and all the transcriptions for replications as well. So to replicate DNA, we start from origin of replication. So regularly we have our DNA double strand, like ladder, right? Now, to get in, we need to peel apart 2 rungs of ladder from each other. So if A and T are together here, we need to peel them apart first, and then we slowly pull apart the zipper. Think about the time when you have a zipper and that broke in the middle, and if you stretch out that middle, the zipper just breaks from the side. That's what we're doing. With the origin of replication and the helicase, make sure to unwind the sides, because right now the DNA ladder is wound very tightly, and helicase helps unwind and makes things easier to access. Binding protein then binds to the starting part near where the initiation wants to go, and then primase starts making the primers, and typically it's RNA primers to get in the beginning, where then DNA polymerase starts to add more nucleotides to a new strand to synthesize a new strand of DNA. Now, if you recall, prokaryotes and eukaryotes have different parts, and in prokaryotes they use DNA polymerase 3, and eukaryotes has A, D, and E for DNA polymerase. And to remove the primers, because originally there are primers on the very top, you don't want a part of your DNA, prokaryotes use DNA polymerase 1, while eukaryotes use RNAs, right? ASE, ASE, think about lyase, right? Things as enzyme, right? RNase wants to get rid of that RNA primer set down in the beginning. Just like how you want to use a ruler in the beginning and later you want to get rid of it. And then RNA is replaced with the DNA parts using polymerase 1 for prokaryotes, and eukaryotes uses polymerase D. Note that there's also DNA ligase to help link things together. Remember, ligase, like glue, lig for glue, and that is to glue the lagging part strands, the Okazaki fragments, together. If you recall, DNA goes from 5 to 3, and there's a leading strand replicating 5 to 3, making a new strand, and it reads from the 3 end. Now the lagging strand will also need to be made from 5 to 3, 5 to 3, 5 to 3, but since it can only go as far as where the zipper can zip up, so it only goes a little bit at a time, little bit at a time, little bit at a time. At the end, the ligase will join them together. Okay, 5 to 3 is the direction for which it will be synthesized. So the first one will be have a phosphate head and then join new parts. That's where it eventually will be. But for now, there's no phosphate here because we haven't added it. You will be adding it in the, where is it? In the Golgi apparatus. That's right. Then at the end, we have topoisomerase to help relieve the tension and to spin it back. Then we have telomerase, which is at the end of the DNA, whichever it replicated a little bit, and that ensures the longevity of the DNA molecule. And then we go back to the beginning, if we need to synthesize new DNA. Remember, DNA is freshly synthesized whenever the cell needs to undergo replication. So the DNA synthesis happens in the S phase of the cell cycle, if you recall from chapter 1. Alright, that's basic it for DNA. Now the biotechnology part comes in several sections. So you typically just need to be familiar with PCR. What it is polymerase chain reaction. It's just a way we can use primers, artificially synthesized primers, to bind to certain parts of the DNA and replicates specific parts that we want. So if we want to amplify a gene, we amplify that part by binding RNAs or primers to one end, and that complementary pairs using the base pairing and the knowledge that if we get another part, we can get that exact snippet to replicate, and amplify and study it in greater amounts. There are also gene therapy and other technology practices to remember which one uses what. You can remember the acronym SNOWDROP, which S stands for southern blotting for DNA, N stands for northern blotting for RNA, and then P is protein and is done by western blot. SNOWDROP, if you write it down, SNOWDROP. It matches over by one. Alrighty, and that's it.